Hello everyone, and welcome to today's live broadcast, Accelerating Immuno-Oncology Research with the PanCancer IO 360 Gene Expression Panel, Advanced Data Analysis Service, and the Signature Development, presented by Dr. Sarah Warren, Director of Advanced Applications, Immuno-Oncology at NanoString, and Dr. Patrick Danaher, at Principal Biostatistician at NanoString. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by NanoString. NanoString Technologies is a provider of life science tools for translational research and molecular diagnostics. The company's molecular barcoding technology enables a wide variety of basic research, translational medicine, and in vitro diagnostic applications. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that green Q&A button located at the lower left of your presentation window, type those questions into the box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice that you'll be viewing the presentation in the slide window. To enlarge that window, just click on that screen icon located on the lower right. If you have any trouble hearing or seeing the presentation, click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. Good news, this presentation is educational and thus offers continuing education credits. Click on the button in the bottom left-hand corner and follow the process to obtain your credits. Please join me now in welcoming our presenters, Dr. Sarah Warren and Dr. Patrick Danaher. I will now turn the presentation over to our speakers. Welcome to both of you.
Thank you, Susie, and thank you for the very nice introduction. My name is Sarah Warren. I'm the Director of Advanced Applications at NanoString, and I'm going to spend a few minutes today to talk with you about one of our newest products, which is the PanCancer IO 360 Gene Expression Panel. And as part of this talk, I'm going to explain to you about some of the wonderful features that we've built into the panel and how we think that it can accelerate research in the IO community. And then I'm going to pass the presentation over to my colleague, Dr. Danaher, who's going to then go on to explain and introduce you to some of the advanced data analysis services that we are building in parallel to support this new expression panel. So thanks. With that, I'm going to advance the slides to talk with you a bit about the state of immuno-oncology today. So since you've joined us on for this presentation, I assume that you are familiar with immuno-oncology as a research field and as a field with many clinical applications. And I'm sure you are familiar as well with the cancer immunity cycle, which we're showing up on this first slide. And so this is the concept that is a hypothesis to explain how tumors and the immune system interact to shape the balance between tumor host permissiveness and tumor host response to the cancer, which shapes whether the immune system becomes activated to recognize and eliminate the cancer or whether the tumor is allowed to persist. So as you can see, there's many potential sites of intervention within this tumor immunity cycle, and many new therapies are being developed which may have the potential to disrupt the cancer immunity cycle and to initiate immune responses that lead to ultimate clearance and protection against the tumor. So at NanoString, it is our goal to build research tools which can seek to leverage the power of gene expression profiling to understand how the balance between the tumor and the immune system is existing with any given patient, and then how potential therapies may be altering that balance to shape clinical outcomes. And by using multiplexed gene expression profiling, we believe we can create a complete holistic picture of what's happening within any given patient. So there's a lot of research that has been ongoing into cancer immunotherapy at the moment. This slide is highlighting just a few of the major publications that have come out just in the past year, which really seek to provide different frameworks for understanding, again, how tumor and how immune systems are interacting. And NanoString leverages publications and latest research such as these, along with our own internal research, to build gene expression panels that are going to be highly informative and highly useful for all of our users. And really our ultimate goal here is to not just to provide static gene expression profiles, but we really want to be part of the solution for providing better therapies to patients in need. And we really want to be part of the community which is helping to generate more effective immuno-oncology drugs, and then to understand how those drugs can be most effectively deployed into the right patient populations, and alone or in combination with other therapies to provide the greatest likelihood of clinical benefit. And ultimately what we'd want to do is help direct the right therapies to the right patients, and we believe we can do that using the gene expression profiles and the assays that we are developing here at NanoString. So I want to start off by telling you about one of these assays, which is called our Tumor Inflammation Signature. So this is a signature that we have been developing with our partners at Merck. It is based upon gene expression profiling of pretreatment tumor biopsies. And Merck began this project with the goal of developing a predictive signature to identify patients who were likely to respond to the PD-1 therapy pembrolizumab. So Merck was a longtime NanoString customer. They were using custom gene code sets to generate signatures and prototype gene expression classifications from their patients that they had acquired through clinical trials. And we're seeking to understand what gene expression patterns looked like when the patients were experiencing benefit from pembrolizumab. So they were working on their own, but then they reached out to NanoString to leverage our expertise in diagnostics to really develop an analytical clinical assay that could be used across a variety of tumor settings to identify those patients who were most likely to respond to pembrolizumab. So this slide is just showing you a bit of what the clinical development of the tumor inflammation signature looked like. Merck initially began by looking at a cohort of patients from one of their melanoma trials, where they did a discovery cohort with a very small number of patients and then validated a preliminary signature. And what you can see is that in patients that had a positive clinical response to pembrolizumab, there was an elevated signature here that was observed versus those patients that did not experience clinical benefit. Merck then went on to refine this signature further by looking at other tumor types, and in both the head and neck cancer and the gastric cancer, they again saw that the signature could be predictive of patients with good clinical response. And they refined the signature, they refined the algorithm that supported the signature to continue to develop something that would be even more predictive. And at that point, we joined with them, and NanoString came on and worked to do a series of training exercises to develop an assay that would become the final lockdown clinical test, which would be taken to the FDA in order to be a companion diagnostic for pembrolizumab, and really to seek to characterize the immune biology that was most reflective of responses to pembrolizumab. So that work was summarized in this paper that came out over the summer in Journal of Clinical Investigations. It was led by our colleagues at Merck, and it describes very briefly the clinical development, which I told you about just a minute ago, and then goes into much more detail about how this signature performs in a variety of clinical settings. I would encourage you to look up this paper and read it and understand it, and reach out to us, please, if you have any questions about how the signature could be applied to your work. Let me tell you a little bit more about the biology of the tumor inflammation signature. It's an 18-gene signature which is measuring the presence of a peripherally suppressed adaptive immune response within the tumor. And that's a lot of words, so let me explain in more detail what that means. The signature itself is a way of determining whether tumors have an inflamed phenotype, so whether they're immune hot or immune cold. It's doing this by analyzing 4 different areas of biology, and you can see those listed on the right side of the slide. So we're really asking 4 key questions. First, are antigen-presenting cells present within the tumor? And if yes, also are T and NK cells, are effector cells present here in the tumor? And then if both those cells are present, we're also looking then for evidence that the T cells and the NK cells have become activated by looking for genes associated with interferon biology. And finally, at the same time, we're measuring for genes which are indicative of T cell exhaustion. So, and this is the sum of these genes is identifying a patient population that may be likely to respond to PD-1 checkpoint blockade. The tumor inflammation signature is measuring the immune genes, so it's not measuring tumor intrinsic gene expression patterns. And as such, it makes it a pan-cancer tumor signature, which means that the same test can be applied across many different indications. And in work that we are doing and work that is ongoing at Merck, the tumor inflammation signature is being investigated in comparison to other biomarkers which are already in the clinic or in clinical development right now. And we've seen very good responses with comparing the TIS to PD-L1 immunohistochemistry as well as mutational load profiling. So I want to start off by showing you just one example of how the tumor inflammation signature can be used to identify patients who may be good candidates for PD-1 checkpoint blockade. This is a figure from the Ayers paper where we're looking across many different tumor types to understand how clinical responses, and in this case it's progression-free survival, correlates with the tumor inflammation signature. So the tumor inflammation signature score is listed across the x-axis here. In this paper it was called the gene expression profile, or GEP. And progression-free survival is indicated on the y-axis. And what you can see is that we've color-coded the patients here based upon their clinical response. Those patients that had very good clinical response based on radiographic signals are indicated in red squares. Those patients that had intermediate responses are in blue triangles, and those patients with poor clinical outcomes are indicated in black circles. And you can see that the data really segregate out into three different areas on this graph. Those patients in the upper right-hand quadrant are the patients who had a very high TIS score and also experienced very prolonged clinical response after receiving pembrolizumab monotherapy. So this is telling us that these are the patients for whom pembrolizumab was the right drug in this setting. In contrast, those patients that are on the left side of the graph are those patients who had a very low TIS score and who also didn't benefit when they received pembro. So these are the patients who had a lack of an immune response already in their tumors. So treating them with PD-1 checkpoint blockade was not enough to overcome that immune suppression. These patients may need a different course of therapy to help them activate immune responses and recruit immune cells to the tumor. And we believe that by looking at the gene expression profiles of these particular patients, we may be able to understand where that particular immune defect is occurring. Finally, the patients on the lower right side of the graph are those patients that had a high tumor inflammation signature but did not experience clinical benefit from PD-1 checkpoint blockade. And so these are the patients who may have more than one mechanism of immune evasion present and active within their tumors. And they may be good candidates for a combination immunotherapy approach, which can overcome these multiple different blockades at the same point. And again, we believe that by looking at the gene expression profiles, we can understand how these different patients should be directed towards appropriate therapies. I mentioned before that the tumor inflammation signature was a pan-tumor assay, but I wanted to provide some data to support that assertion. So this is looking at the tumor inflammation signature from 11 different tumor types. And each tumor type is indicated across the top of this graph by a different color. And you can see that we've organized these samples from the TIS low to the TIS highest scores going left to right here. And there is not an intrinsic clustering of the different tumor types to one region of TIS score versus the other. So this is telling us that, again, as we believed by looking at the biology, the tumor inflammation signature is really measuring the fundamental immune response within the tumor rather than gene expression coming from the tumor cells themselves. And finally, I mentioned as well that we are comparing the tumor inflammation signature to other potential biomarkers. And so one of the reasons that we really believe in the power of the TIS is that it's using the gene expression profiling to directly interrogate the immune biology within the tumor. We have preliminary evidence that shows that this test may have better predictive power than PD-1 immunohistochemistry. And we are currently developing this now as a clinical trials assay, which may be deployed into registrational trials. So this has been developed very rigorously under GLP conditions for use in investigational research settings. And finally, since it's based upon the NanoString platform, this is a decentralized model. So this assay can be run wherever there is an nCounter instrument installed and in place. In contrast to the tumor inflammation signature, other biomarkers which are currently in development for use as predictors for immunotherapy have some significant problems with them as assays. So the one that is approved as a diagnostic right now is the PD-1 immunohistochemistry assay. This is a single biomarker analyte, a single analyte biomarker, which is just measuring the presence and localization of the PD-1 protein within a tumor biopsy. And the PD-1 protein is known to be very diversely regulated with regards to its spatial profiling. It is regulated temporally. It is very dispersed in terms of the distribution that you see when you look at patient samples. And consequently, the PD-1 assay has very low predictive power in terms of identifying patients that will respond to checkpoint blockade. In addition to variations intrinsic to the biology of PD-1 expression, since this is an immunohistochemistry assay, it's relying upon, at the end of the day, a subjective interpretation of that test to appropriately assign the patient to a given score and then potentially a given therapeutic course of treatment. The other assay which is in clinical development now is to measure a variety of assays to measure tumor mutational burden. And this is an intriguing hypothesis for developing new biomarkers. Unfortunately, the tumor mutation burden is sort of intrinsically limited because it's, again, a single measurement of one facet of tumor biology. So it's really looking for the presence of potential neoantigens within a tumor rather than looking at the downstream effects of actual immune activation within that same tumor. And finally, it has, as it is currently being developed right now, it is a centralized model of performing the assay, which brings in logistical challenges to deploying this assay and multi-site clinical trials. And finally, since tumor mutation burden is just measuring the total mutational load, it has no abilities then to predict varying responses to combination therapies, where you might expect a more complex signature like gene expression profiling to give you additional predictive power in that setting. I wanted to show one slide next that you're seeing here that compares the tumor inflammation signature to the PD-1 immunohistochemistry. And so this is an assay, this is a test that was done in head and neck cancers where patients' tumors were scored by the TIS assay and also by the PD-1 assay. And those results from each of those tests were then compared to the clinical benefit that was observed for those patients. And what you can see here is that the tumor inflammation signature has better predictive power than the PD-1 assay. It's actually has higher sensitivity in this test. And the area under the curve is greater, suggesting that it has the greater predictive power and better able to identify patients that will respond. Similarly, when we compare the TIS to tumor mutation burden, again in the head and neck cancer setting, we see a very interesting result. And head and neck cancer occurs from one of two causes. There are either those patients who are positive for the HPV virus and whose tumors arise and tend to have very low mutational burden. And in these patients, as we're seeing on the left side of this graph, the tumor inflammation signature has a very high predictive power because it is measuring the presence of the active immune system within that tumor rather than the potential immune activation as would be indicated by looking just at mutational burden alone. Similarly, the other cause of head and neck cancer is through exposure to carcinogenic agents. And in those patients, then the tumors that arise are negative for the HPV virus. These patients tend to have higher mutation loads within their tumors. And in this case, we see predictive power from both the tumor inflammation signature as well as the mutational load profiling. And so what this tells us here is that in some cases, mutation load profiling can be predictive in the clinical setting. But in all cases here, we're seeing that measuring the actual biology by looking at the TIS, we're able to enrich for those patients who are likely to then respond to PD-1 checkpoint blockade. So I'd like to tell you then now about a new panel that we are developing that can be used to build upon the tumor inflammation signature and more deeply interrogate the biology of tumor-immune interactions. And just to remind you that this panel, as for all of our panels, is working on the NanoString platform where we're using the targeted capture and reporter probes that hybridize specifically to the RNA from the sample of interest, and then reading those out in a digital format so that we're getting, at the end of the day, precise quantitative counts that are reflective of the presence of that particular analyte within the sample that is being interrogated. And we build these panels out to enable interrogation of up to 800 targets at once. And our goal with building additional gene panels is always to enable our customers to answer more refined biological questions. And that is exactly what we wanted to do in this setting as well. So we wanted to leverage the power that we've been developing around the tumor inflammation signature as a clinical assay and build out a research use only platform that could then be used to further interrogate mechanisms of immune evasion. And the result of this effort was a product that we call the PanCancer IO 360 gene expression panel. And the goal of this assay is really to provide an actionable profile of tumor immune interactions that can then help guide research and development around novel immunotherapies. And so the way that this assay works is that it provides 800 gene expression profiles of patient samples, in these cases pretreatment tumor biopsies, and we have incorporated on top of these a series of signatures, including our tumor inflammation signature, that can help dissect the immune pathways that are present within this particular sample, and then ultimately, be able to link those pathways back to particular therapies, which will induce positive clinical outcomes for those patients. So the IO 360 panel has 770 genes built in and pre-designed. We also have space open for 30 genes to be included as a customized panel spike-in. We've included 20 reference genes in this panel, and the panel itself has content that addresses both the tumor-specific signaling, the tumor microenvironment, and the immune response. And within this panel, we have specifically developed individual signatures and pathways to characterize the different biological activities that will allow researchers to very quickly understand and at a very deep level ask questions about the particular interactions between the immune system and the tumor that are being interrogated in this research setting. So we've provided annotations about each of the genes that are in these panels. We've also provided the signatures as well as particular subsets of signatures that enable immune cell profiling. And I'm going to give this presentation over to my colleague, Dr. Danaher, in just a few minutes, who will tell you more about what each of those signatures are and what they can do. But just to give you a very brief overview of what these the kinds of biology that these signatures are addressing, you can see here the gene content that we have incorporated that interrogates in a very holistic fashion signaling patterns that can shape how tumors and immune systems are interacting with each other. And this is just a list of those particular signatures that have been embedded into the panel and which are available through our data analysis services. So when you leverage the IO360 panel in your research, you get the gene expression panel itself. It's able to be analyzed with the NSolver software, as all of our other gene expression panels are. And then we've built these signatures in as an added level of functionality to really help investigators ask the more detailed and more refined questions about their samples. This is just very briefly one paper which describes the cell type profiling signatures that are embedded in. And just as an example, we can use this panel, we can use these signatures then to understand how relative levels of immune infiltrate vary from one sample to the next. And I'm going to very briefly mention the data analysis service options. So as I said, all of the annotations are available for this panel to be used, and samples can be analyzed through the NSolver and the advanced analysis software packages. And then we've also built additional data analysis services where we can calculate the precise signatures that are embedded within these panels and provide back to users a single sample and multi-sample reports, which allow you to interact with the data in a very dynamic and interactive way and very quickly generate new hypotheses and quickly advance the research in immuno-oncology. And so as a result, I believe what we've built here is a system which enables very easy and very robust profiling of tumor tissues to enable discovery of new biology, validation of new hypotheses, and creation of new biomarker signatures that may be useful for directing patients to the appropriate immunotherapy. The core of this IO360 panel is called the Tumor Inflammation Signature. It is measuring the existing adaptive immune response within the tumors, and it performs very well when compared to immunohistochemistry and mutational load. And then in addition to the tumor inflammation signature, we have also built additional signatures around the panel, which can help you ask even more detailed questions about the biology of your sample. And with that, I'm going to turn the presentation over to Dr. Danaher, who's going to tell you more about what these signatures are and what they can do. Thank you.
Thank you, Sarah. So I'll describe briefly, let's see, both all the work that's gone into creating this analysis service and then what the analysis service can bring to any particular data set you might have. So I guess the goal of the analysis service is just to bring understanding to immuno-oncology gene expression data. Having a data set with 800 genes and 10 or 100 samples is it's a complex matrix of a lot of numbers, and it's hard to extract a lot of meaning from that, especially if you're not a statistician or if your informaticist has a lot of projects in this pipeline, which seems to be the state of affairs. So yeah, we just want to make it easy to extract not only information but understanding from your data. So I guess the first question is just, given a sample, given a tumor, what do you want to know about that sample? The first question, of course, is just, is it immune hot or cold? So that would be the simple TIS. But then there's a lot of other things we'd want to learn about the tumor. I would want to know what cell types are present, what immune cell types are present in the tumor and how abundant they are. And really, we want to know every detail of the cancer immunity cycle as we currently understand it. How foreign does the tumor appear? Is it accessible to the immune system, or are there structural barriers? The immune system that is in the tumor, what are those cells doing? Are they exhausted, or are they actively cytotoxic? And then how is the tumor responding to the immune system? Is it expressing a lot of checkpoint and other inhibitory molecules, or is it growing? Is it dying? Is it responding to interferon signaling? All questions that we understand the biology, and it would be really nice if we could measure them in some way. So a big part of this, the analysis service, is creating tools to measure the variables we care about, everything I just talked about. So the first thing to begin with, Sarah mentioned all these signatures we've generated. And I think it's worth describing a little bit how we actually derive these signatures. There are probably more gene signatures that have been published than actual genes in the genome, so it deserves a bit of explanation why these signatures are actually useful and deserve attention. Let's see, so we've got a pretty rigorous process and a pretty selective process. We attempt signatures for a great many biological variables, and we actually fail our signatures in a good percentage of the time just for not having strong enough evidence. So the basic outline of our process is as follows. We begin with known biology. If we want to measure, say, cytotoxic activity, we use biological knowledge, lit searches to come up with a gene list that is likely to be mechanistically involved in cytotoxic activity. So, just from the get-go, it's very plausible that expression of any of these genes ought to measure cytotoxic activity. But, of course, we're not going to be right all the time. Many genes are regulated after, they're regulated at the protein level or are influenced by a number of factors, not just the activity we want to measure. And so, many of our candidate genes just aren't going to measure, whatever biological process we care about. Or many of our cytotoxicity genes, for example, might actually just be expressed at constant levels in all CD8 cells, just ready to be activated at the protein level. And so, we think we're measuring cytotoxicity with these genes, but actually we're just looking at CD8 T cells. So there's a lot of ways we can go wrong if we just stick to biologically derived gene lists. So we take a second step after using the literature to come up with a candidate gene list. We use co-expression analysis in TCGA to winnow down our gene list, to test that the genes are performing as they ought to if they are indeed measuring what we think they are. And I'll go more into that. But the basic idea is we define ahead of time, okay, if these genes are actually measuring cytotoxicity, what co-expression patterns should they display? And then we can just look at their co-expression patterns in TCGA, see how well they adhere to the pattern we expect, and we can remove genes or entire gene lists as needed based on how well the data agrees with our predefined model. So to make this a little more concrete, let me start with a failed example of a signature. We wanted to create a signature of T-cell co-inhibitory signaling. You might expect there could be some variable of how receptive T cells are to co-inhibitory signaling and how much co-inhibitory signaling is actively affecting those T cells. So we chose, using the literature, a good number of co-inhibitory genes. I'm just showing 4 of them here. And you'd expect that if these 4 candidate genes all measured co-inhibitory signaling, that they would be pretty correlated with each other. Is that biological process went up or down, it would drive all those genes to go up or down, and that would induce correlation amongst them. And if we look at the normalized data, this is TCGA melanoma. The plot on the left here, you can see that just normalized gene expression data, these genes do correlate quite well with each other. They appear to be measuring the same thing. But we're not satisfied with that. We apply a few more conditions. We want to make sure that these genes are measuring what we think they're measuring and not just some other variable that happens to drive their co-expression. So we take the normalized data and we regress each of these genes on a number of other factors that we are worried might influence their expression. So here an obvious factor is T cell abundance. We have a signature for that, so we regress these genes on that, as well as a number of other variables we're concerned about. And then we look at the residuals of those genes from the regressions. And if we look at the plot on the right now, we can see that once we regress out all these other signatures we were worried about, no co-expression amongst these genes remains. So while these 4 genes might be measuring the same thing, that thing is not some independent variable of T cell co-inhibitory signaling. The thing they're measuring is probably just T cell abundance. So this is the case of a failed gene signature. For a successful case, we wanted to look at antigen presentation by the MHC class II. So here again are just 4 of the genes we looked at. In the normalized data, we see they're highly correlated. So, they're definitely measuring the same thing. It's an encouraging start. But we're not satisfied with that, so we regress on 10 other variables. We regress on signatures for the abundance of a number of antigen presenting cell populations, as well as other variables that we know broadly influence gene expression. The amount of proliferation in a tumor, general signatures of lymphoid and myeloid abundance, stroma signature, and then just for good measure the first 3 principal components of the data. And what we see is after conditioning on these 10 pretty powerful variables, that explain quite a lot of the transcriptome, on the right we see that much of the correlation amongst these 4 genes remains. So at this point I think it's fair to say that these 4 genes are measuring the same thing, And that thing is probably antigen presentation by the MHC class II and not some other factor like B cell abundance or dendritic cell abundance. So this is the kind of evidence we demand of our gene lists before we can combine them into a signature. All right, so with that background out of the way, I'll proceed to show with an example of how these signatures can help you understand the data set and get a little more statistical power to boot. So this data set is tumors treated with a checkpoint inhibitor. We've got 34, it's a small data set, just 34 non-responders and just 8 responders. So we're not spectacularly powered here. In this data set, we're actually looking at almost 2,000 genes instead of just the 800 in our panel. If the panel, unfortunately, wasn't available at the time. Let's see. Yeah, so the first thing to point out is just that when you look at signatures instead of single genes, just by the simple fact of cutting the number of variables you're looking at, you gain a lot of statistical power. Now, I'm assuming that this proposition only works if the signatures are actually relevant to the biology in question, which they are in this case. So if we look at the top 2 plots here, I'm just doing differential expression of genes and signatures. The volcano plot for genes is on the top right, for signatures is on the top left. So if you're unfamiliar with these plots, horizontal axis shows differential expression between responders and non-responders. Vertical axis shows statistical significance, with everything above the dotted line being a 0.05 p-value or better. So if we look at the signatures, we see that many are significant. A good number fall above 0.05. And if we look at the genes, a whole ton are really significant. Some pretty tiny p-values there, some below 0.001. So there's a lot of information in both cases here. But of course, we can't just test 2,000 hypotheses and be satisfied with the p-value cutoff of 0.05. We need to do some sort of adjustment for multiplicity to make sure we're not getting excited about genes just because they were lucky instead of actually useful. So we perform a false discovery rate correction and obtain the volcano plots on the bottom. If you look at the signatures, now plotting FDR on the vertical axis, false discovery rate instead of p-value, we find that many of the signatures end up with a false discovery rate just around 0.05. It happened to be a little bit below, but that's not really the point here. The point is about half our signatures, we've got a pretty strong conclusion to be made. We're not 100% confident, but we expect that less than 5% of these discoveries will go on to be false. If we look at the bottom right volcano plot, false discovery rate of our single genes, we find that even our strongest p-values, those below 0.001, they only amount to a false discovery rate of about 70%. So we're really left with no high confidence conclusions here. And this is just the basic fact of working with high-dimensional data in a small study. Unless you have a truly spectacular effect size, you're never going to see anything with high confidence. But in this study, if we had stuck with genes, we would have been unable to make high confidence conclusions. We'd have just been able to say, there's some interesting genes, but we'll just need to collect more data to see if they validate. If we begin our analysis by focusing on signatures on this smaller dimension but still highly useful set of variables, we actually end up with some confident conclusions. Another use of the signatures is not statistical power, it's just simple understanding your data. There's a lot of genes, and many of us know what many of them do, but it's still pretty confusing looking at differential expression results for hundreds of genes, maybe half of which you've heard of if you've been working in the field a while. On the right now, I'm just showing how all of our signatures differ between responders and non-responders in a forest plot. So the box is the estimate, and the line is a confidence interval for the effect size. So everything to the right is upregulated responders versus non-responders. And you can see at a pretty quick glance that there are quite a few signatures that are upregulated in responders. And looking at them, they're generally the signatures involved in adaptive immunity. So largely members of the lymphoid compartment and their associated activities. So CD8 cells or cytotoxic activity or interferon gamma signaling, that sort of variable. Variables, signatures associated with the myeloid compartment, like dendritic cells, macrophages, neutrophils, those are generally not different, or on average are not different between responders and non-responders. So they don't appear to be informative for that distinction here. And then interestingly, arginase is that one point out on the left. It's the single variable we're looking at that's upregulated in non-responders. So perhaps it's an important inhibitory factor here. So these signatures are interesting individually, but they're also in combination with each other. So looking here, I'm just plotting 3 interesting signatures against the tumor inflammation signature. I think an important point is that many immune variables are pretty correlated with each other. As the immune system goes into a tumor, it all goes in, both the CD8 cells and many, many other cell populations as well. So it can often be hard to pull out information from this tight correlation. So we find that just plotting variables or regression variables against the TIS allows us to see in a little more detail what they're actually doing. So this plot on the top left, looking at macrophages versus TIS, I thought was pretty interesting. Here the responders are in red, non-responders are in blue. You can see that to be a responder, you have to have a high TIS. All the red points are on the right side of the graph. But if you have a high TIS, it looks like it's helpful to actually have lower macrophages rather than higher macrophages. It's on the border of statistical significance, not quite there, but it's an interesting result that deserves more follow-up. You might expect that these macrophages are performing as myeloid-derived suppressor cells in these samples. And so highly immune active sample, an immune hot sample with a high TIS that also had a lot of macrophages, that immunosuppressive axis of MDSCs might not be adequately dealt with by this checkpoint drug, and so that sample would remain a non-responder. If we looked at macrophages alone, just that one variable one, we wouldn't see this. The average macrophage score is the same between responders and non-responders. But after conditioning on the TIS, it actually becomes interesting. And finally, I want to show how these signatures can be used to understand even a single sample. So of this data set, I've picked out what I think is the most interesting sample. It's a very high tumor inflammation signature score, so, a very immune hot tumor that nonetheless didn't respond. So it bears asking, this tumor is immune hot, what kept it from being pushed into successful anti-tumor immunity by this checkpoint inhibitor? So we can look at these signatures and try to see how this tumor looks in them. So let's start at the top right with antigen availability. I look at signatures for antigen presentation machinery, for the immunoproteasome, for destruction of the antigen presentation machinery, or for mismatch repair deficiency. And looking at these top 4 signatures, I see very high levels of antigen presentation, no degradation of the presentation machinery, and possibly some evidence for some mismatch repair deregulation. So if anything, this tumor is highly visible to the immune system. It's presenting, it's very actively presenting antigens. So nothing here to hold back an immune response. Looking on the next row at variables involved in anti-tumor immunity, again, we see very high, really encouraging values of highly abundant CD8 cells, a lot of cytotoxic activity, and a lot of interferon gamma signaling. This is a fully functioning, highly active immune response. Everything here suggests this patient should have responded as well. And then on the bottom right, we can look at inhibitory factors. And all these various checkpoint or other inhibitory molecules, they're high, but they're actually not high conditional on the patient's tumor inflammation score. Just looking at PD-L1 versus TIS, for example. PD-L1 is quite high, but all samples with high TIS have high PD-L1. So we shouldn't be particularly concerned about this patient's response based on the high PD-L1. So we don't find an explanation here. Where we do get an answer, I think, is in looking at the arginase expression of this patient. Looking at arginase in general, I see 2 clusters. There's a cluster which has basically no expression along the bottom there, and then this more diffuse cluster which has moderate to very high expression. And this sample had particularly high expression of arginase. And of course, we saw earlier that arginase was quite predictive of response versus non-response. So if I was to explain this patient, I would say that metabolic factors that arginase is involved in are probably what's holding back the response here. So a major goal of a lot of studies in the translational setting is to actually not just understand the biology behind response or non-response, but to create a predictor of response, to have a diagnostic that could exclude non-responders from a clinical trial, for example, or identify the patients where your drug is going to benefit patients more than the standard of care. So in this case, we also find that our signatures end up contributing statistical power and enabling success. So this is a simple, not quite simulation study. I took the same data set and I split it into 100 training and test sets. In each training set, I trained a signature either, I trained a predictor of response, either using only genes or only signature scores or both. And then I tested that signature's predictiveness in the data I had left out. So we've got 100 different attempts to train a signature and then independently validate it. And what I'm plotting here is the test set area under the ROC curve of each of those 100 or 300 trained signatures. So, remember when we look at an ROC curve, area under the curve of 0.5 means you have a random predictor, and area under the curve of 1 means you have a perfect predictor. So what we see here, if we train using only genes, our test set area under the curve is pretty weak and pretty variable. Sometimes it's even worse than random. If we train using signatures, the average performance goes up substantially from around 0.55 to about 0.75, so a pretty huge jump in performance. And the key here is just we don't have a very big data set. So machine learning using 2,000 features from only 40 or so samples is just, it's a hard battle. No matter how sophisticated the machine learning method, that dimension to sample size ratio is just going to create a lot of problems for it. Whereas if we can, if we can focus on the variables that matter, the variables that we already expect are going to be relevant to the problem that we've tried to capture with these signatures. And if we can look at fewer variables, we can give ourselves a lot of statistical power. So our data analysis service has a unique method for trying to optimally combine genes and signatures to create predictors of response. We don't want to wholly lean on our signatures, but we think we can add a lot of value by heavily weighting them over the genes. Another thing we do with this analysis service, we try for each sample to identify, for each given sample, what are the factors holding back the immune response? Why is the immune system not already killed this tumor? And the hope is, one day that we'll have an amelioration for each of these factors. That you can imagine some future tumor being assayed, and we'll see that it's got these two high inhibitory factors, and it's lagging in this immunoactivating factors. So let's address each of those three factors. As an example of how this works, the plot on the top right, we see this tumor had very high interleukin-10, and so it's an inhibitory factor. So that might be, that unusually high expression might be a problem for this patient. That might be holding back their immune response. The middle plot, we see very high CTLA-4, and actually this is kind of interesting. Normally CTLA-4 tracks TIS, but in melanomas it seems that CTLA-4 has this other cluster, very high CTLA-4, despite being otherwise immune cold. And this is one of those patients, and I think CTLA-4 expression is a pretty good explanation for why this particular tumor has been able to evade the immune system. And finally, we also look for signatures that are immune-activating signatures that are lagging behind the rest of the immune response. So this sample on the bottom right is very low, I guess it is kind of a middle-of-the-road antigen presentation machinery signature. But conditional on the rest of its immune activity, it's really low. Conditional on its very high TIS, that is, it's very low. So what seems to be happening in this sample, there's high immune activity, lots of T cells and CD8 T cells, cytotoxicity, interferon gamma signaling, but for some reason the antigen presentation machinery is just not keeping up with that. So it might be that the tumor is somehow downregulated that, downregulated B2M, for example, to try to evade immune detection. And this is the kind of barrier that is probably pretty hard to treat, and might make this unpromising tumor to treat with immunotherapy despite its immune hot status. Finally, independent of all the signatures, there's more the gene expression can say about the tumor's immune state. There was a fascinating paper recently by Patel and Restefo, which used CRISPR screens to identify genes that are essential for response to immunotherapy. And they found 30 or so genes that, if disabled, rendered a tumor resistant to immunotherapy. And it turns out that a lot of these genes, you can identify many outlier low levels of them, many loss of expression events, just looking at primary tumors and TCGA. So in these top two examples, SOX10 and MLANA, you can see in one case infrequent, and in another case fairly abundant instances of loss of expression. And I think it would be a reasonable hypothesis that these loss of expression tumors are less promising responders to immunotherapy. Though, of course, that has to be tested, but it's always good to be able to identify these events when they happen. Other variables are a little more, little harder to pick out, though we can do so. B2M is probably the most famous gene that's required for response to checkpoint inhibition. And basically, B2M has a really wide expression range, so it's hard to say if a particular sample is unusually low B2M expression. But if we plot B2M against other antigen-presenting genes, we can actually resolve some of that, or we can condense some of that variability. So looking at the bottom right plot, there are many samples that have middle-of-the-road B2M. Take this sample, for example. It's got pretty average levels of B2M, but conditional on the rest of its antigen presentation machinery, that's really low. It seems like B2, this is on the log 2 scale, so that's about 8-fold downregulated from where we would expect it to be, as is this sample. So in these cases, it seems like B2M has been drastically downregulated, possibly in a adaptation to help the tumor avoid the immune system. So just another variable that we can use to describe what's happening in the tumor-immune interface using gene expression. All right, well, that's all I have to share there. As Sarah said, we're actively developing this analysis service. We're constantly adding variables to it, new signatures, new analyses as we discover them. And yeah, our goal is just to take a complex data set and make it understandable, render it into the variables that we understand as biologists rather than as computational experts. Thank you very much, and I think we're happy to take questions now.
Thank you, Dr. Warren, Dr. Danaher, for that informative presentation and for your important research. We now will start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of the presentation window, type your questions into the box that appear on the screen, and click the send button. We'll answer as many questions as we have time for. So let's take a look at our audience questions already coming in. And thank you, audience, for your live participation. Our first question is the TIS likely to predict response to other immune checkpoint therapies? And either one of you can go ahead and answer that.
Yeah, I can take that answer. Thank you very much. That's a really good question, right? The TIS was designed to measure the inflamed tumor state, and as such, we are very hopeful that it will be able to predict other immune therapies. We are currently under investigations that will allow us to answer this question with greater detail, and we look forward to sharing these data with you as they become available. We will have some of this data available at SITC, and we are continuing to develop and publish other data sets that will help us answer this question.
Thank you, Dr. Warren. And here's our next question. Are there any advantages to using gene expression-based assays as biomarkers versus other conventional technologies?
Sure, I can take that one as well. So gene expression assays are a very powerful tool for understanding the fundamental biology of the system that you're studying. Because we can leverage the highly quantitative nature of the NanoString platform to create these gene expression profiles, and because we can look in multiplexed gene expression space, it allows us to create more complex and more nuanced predictive signatures that can help identify patients who may be more likely to respond to one therapy versus another. And so that can help us to avoid some of the heterogeneity that is introduced into other samples based upon variable expression of a single biomarker readout. And it can also allow us to begin to, for instance, segregate out patients who would respond better to a combination therapy from a monotherapy. And those are two aspects of signature development that other assays currently in development are not able to address today.
Thank you. We have time for one more question. And again, I want to thank our audience and let them know that our speakers today are willing to take further emailed questions, and they will email their responses. Our last question that we have time for is the TIS relevant for indicating patients to novel immunotherapies versus single agent anti-PD-1 or PD-L1?
I can take this one, thanks. Yes, that's a really good question. So when I see that, when I look at data from a small clinical trial and I see that response to a therapy is correlated with the tumor inflammation signature, I say, great, you're enriched for responses in hot tumors. You know, we can improve the power of your trial because we can exclude the immune cold tumors and get rid of a bunch of non-responders while holding on to the responders. The other situation, of course, is that we see immunotherapy in which response is not correlated with the TIS. In which case, I say this is really great. We can. It looks like your drug works just as well in immune cold patients as in immune hot patients. In immune cold patients, you've got a really strong argument for using this drug or this combination versus single agent checkpoint inhibition. Whereas in immune hot patients, you know, maybe this extra combination, this newer drug, isn't going to have much of a performance benefit against, say, pembrolizumab or nivolumab. So, I guess whatever the case is, you can either you want to know both which patients is my drug going to help, and secondly, which patients will my drug help versus the single agent standard of care. And however you perform across the immune hot cold spectrum, there's information to be gained from the tumor inflammation signature. And then I would hope lots of information to be gained from these other signatures. You know, there are diverse mechanisms of resistance to immunotherapy, and we think we can capture a great many of them with gene expression.
Wonderful. Thank you both. Did either one of you or both of you would like to make a closing remark before we end our webinar today?
Sure. No, I just wanted to say thank you very much to the audience for joining us for the last hour. We hope that you found the information that we presented informative and intriguing for your research. If it's something you would like to explore more, I would encourage you to go to our website, nanostring.com, where you can find more information about the IO360 panel, the tumor inflammation signature, and some of the data analysis services that we have available. And there are contact pages throughout the website that can put you in touch with our scientists and our representatives at the company who can help you learn how to bring NanoString profiling into your research. Thank you.
And I want to thank you both again for your important research and your presentation. I'd also like to thank LabRoots and our sponsor, NanoString, for making today's educational webcast possible. Audience members, before we go, I want to remind everyone that today's webcast will be available for on-demand viewing through February 2018. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks for joining us. We hope to see you again soon. Goodbye.